to next call up Slava Karolis from King's College London, speaking on Fetal MRI dataset of the developing Human Connectome Project, a window at the earliest stages of brain connectivity and development. Thank you.
Thank you so much. Hold on. Is it mouse control too?
This would be forward.
Okay. Okay, thanks so much. Okay, so I'm here to give an outline of our fetal MRI dataset on behalf of whole Human Connectome Project. So I have no disclosures to reveal. So let me start with a few words about the project itself, because I have impression some people confuse it with Human Connectome Project and its derivatives. It's actually not related project and was funded by European Research Council, and the grant was awarded to three British institutions: Imperial College, University of Oxford and King's College of London. And the goal of this project globally was to provide an open access and highest possible quality data for studying brain connectivity development in the perinatal period. We had a few releases, and recently in December we finally released our fetal dataset, which I'm going to talk about it, spanning the period between 21 and 38 weeks in utero. So during the fetal period, the brain undergoes significant changes, most likely more significant than over entire lifespan ex utero, and which makes this period sensitive to adverse effect of prenatal factors, and which may have long-term effect on well-being. Therefore, this motivates studying fetal brain in utero and in vivo, but it's, as you can probably appreciate from this image, it's an incredibly difficult task to do. It's inherently low signal-to-noise ratio data. It has very high level of motion. Non-rigid high-intensity mother tissue also interferes with acquisitions and preprocessing of the data, and many other factors. So our goal was to provide the community with a fetal data set which was acquired with using motion-tolerant acquisition methods for multimodal data, T2 weighted, T1 weighted, diffusion, fMRI, which were acquired in accordance with strict safety constraints, including such meticulous things like position of mother in the scanner. And we release the data in both in raw and preprocessed form. In our release, we tried to mirror, we mirrored the bits formatting, even so the data itself that doesn't fit this format well, but providing this data in the format and also using conventions which are similar to bits format, we thought we can make make the life of the users easier so they can recognize familiar structures in the data. So in the following, I'm going to give an overview of the workflow, the preprocessing stages, the results of these preprocessing stages, and the key applications. And I start with anatomical data, which in our data set consists of 296 sessions from 273 unique subjects. So because there's a high motion levels in fetal data, we applied single shot acquisition sequences which freeze motion time, but then we also sample data in orthogonal planes, which allows us then to use slice to volume reconstruction techniques in order to produce super resolved images in a standard rotation. This was followed by performing tissue parcellation using the OHBM toolbox, which was then, the output of which was then manually corrected, meticulously corrected. And these corrections allowed us then to extract accurate surface representation and associated metrics for this data. Furthermore, these corrections allowed us also to develop improved automated tool, which is also openly available for community to use, which is called Bounty. The richness of data also was useful to produce dHCP special temporal volumetric atlases, which apart from including the anatomical data, also include images of diffusion-derived metrics and group-level tissue parcellation. The same atlas served as a template for registration structure for data fusion and group-level analysis. The key point about this registration infrastructure is that we computed transformation between nearby image spaces, such as a T2 native space and H-match template space, as well as between H-adjacent template spaces. And these building blocks, these transformations then were concatenated in order to provide arbitrary mappings between various spaces, including connection to the neonatal template space. Here is the data, surface data allows us to compute the first comprehensive spatial temporal surface atlas, which spans the period from 21 to 36 weeks. And this atlas already getting attention from the neuroscientific communities. For instance, these two papers were looking at the biological processes, development processes in utero in association with gene expression, and I was told that one of the papers was just accepted for neuron publication. Next, our diffusion, our functional data, which consists of 275 sessions, 350 volumes, acquired over approximately 13 minutes. We take into consideration not only magnitude data but also phase data, because in the context of fetal imaging, the phase data is very useful for estimation of B0 field inhomogeneities dynamically, and which can be then used to perform distortion correction. We also perform slice to volume reconstruction of the data, which showed improved performance over the volumetric alignment. As an exemplary case at the top, where red means beta TSNR in slice to volume reconstructed images, and at the bottom is a comparison of at the group level. We also used a sophisticated regression-based model, including very rich set of denoising regressors, and if you're interested in more details about this data, I'm going to give a talk about this tomorrow. But also we have a preprint very detailing everything about what we did with fMRI data. Naturally, the data is acquired in order to study functional connectivity development in utero, which in itself is also analytical challenge. And in the paper on the right, we proposed an alternative to group ICA methods, which we think may be more suitable for the data when we observe such huge maturational changes over a short span of time. Last but not least, our diffusion data, which is a multi-shell hard disk sequence, which was acquired such that diffusion gradients and strengths were optimized empirically. We also take care of the fact that diffusion gradients prevents accurate estimation of phase offset, and therefore our sequence allows us to actually compensate for that. And then we start with denoising the data. We next moves to performing dynamic distortion correction, and at the last step we use a shard pipeline which takes into account the variation in diffusion encoding due to variable position of the fetal brain. So the method basically iterates between slice to volume reconstruction and model prediction with outlier rejection in order to accurately model diffusion signal, and in the end we able to estimate orientation distribution functions. So the applications of our diffusion data are shown in these two papers, which shows you that we're not only able to reconstruct biologically plausible connectivity tracks, white matter tracks in the brain, but also using the fact that our acquisition sequence is multi-shell, we can also model macrostructural properties of the brain. So here is the links to our release data, which is with NDA, also dHCP atlas you may find interesting, useful to use. And you also may be interested to consult our website, which at the moment undergoes major refurbishment, but still we got some information there, including how to download the data from NDA. But also one thing which I didn't mention is that we also have a wealth of non-imaging data, which you might find useful. Obviously, it's a huge project where many people contributed, and even beyond PIs, we've got a group of group leaders who led this project in many ways. But I also would like to single out a few personnel here, from the personnel here, because they are critical for making this fetal release happening. And because perhaps people here are interested in developing brain development, I would like to use this platform to mention that there will be soon the announcement of special issue in brain instruction and functioning and would suggest maybe you could consider it as an outlet for your publications. And thank you.
We have time for a question, and I apologize, I missed some of the electronic questions in the prior speaker. Maybe at the end we'll be able to come back to those. But any live questions at the moment? Oh, here comes a live question.
Hello. Thank you. Very interesting data set. I just wonder, is there any plan for some follow-up, more acquisition of these subjects to make some sort of a subset of longitudinal data as well?
There's plans, but some researchers from our departments submitting applications, and they need to convince people to fund this. So far, there's no money for that, as far as I know.
It would be amazing. Yeah. Thank you.
If no more questions, we'll start the GoFundMe site for the next acquisition, and thank you.